Today we've got a speaker coming to talk to us. He's Dr. Robert Casper. His area of expertise, one of them, is stem cell research. He's a world-leading researcher in reproductive sciences. He holds the Camille Dan Family Research Chair in Translational Cell Biology. He's a professor in the Division of Reproductive Sciences in the Department of Obstetrics and Gynecology at U of T. He also helped to begin Canada's first cord blood bank. Much of Dr. Casper's research involves use of stem cells. Some key areas of his research include using umbilical cord stem cell technology for potential treatments in cardiovascular disease, diabetes, neurological disorders, and using infertility treatments. He also happens to be the father of two Georgians, one of whom is over there. So please join me in welcoming Dr. Robert Casper.
Thanks a lot, Mr. Spacey. Good morning, everybody. It's a pleasure for me to be here, and I hope I got you out of something you maybe didn't want to be in this morning, too. So I'm going to talk about stem cells. You probably have heard a lot about stem cells in the news, especially for treating things like brain injury and replacing organs and things like that. And Michael J. Fox is one example. He has Parkinson's disease. That means he's missing some of the neurons in his brain that produce dopamine. It's a neurotransmitter. And they get tremor and rigidity, which you see when you see them on film now. Anyway, most people, when they think about stem cells, are thinking about tissue regeneration like this. And there are two types of stem cells. One is embryonic stem cells, and the other type is adult stem cells. Everybody's really thinking about embryonic stem cells. Those are the ones that have been in the news. Those cells, as the name implies, are derived from a human embryo. So this is a picture of a human embryo at day 5 after fertilization. It's called a blastocyst. And at this stage, this part of the cell right here becomes the baby. And these cells around the outside become the placenta. So this is if you picture like a basketball, the inner cell mass would be where the writing is on the top, and all the rest of the sphere is made up of the cells that become the placenta. So embryonic stem cells are derived from this area right here, the part that becomes the baby. So those cells are actually taken out of the embryo. They're grown in a dish. And one of the advantages of embryonic stem cells is that you can just keep growing them. They're immortalized cells, so they just grow forever. But also, if you add certain chemicals, there are plenty of recipes now that have been developed over the last 20 years. You can actually get these cells to divide or differentiate into various tissues, like muscle or nerve tissue. Okay? So this all looks great. These cells are very impressive in what they can do. But there are some problems, and the first one you can see right on this slide is that you have to destroy a human embryo in order to get these cells. So there's some ethical and religious issues related to the development of embryonic stem cells. Many countries, including the United States, won't allow research on embryonic stem cells for that reason, because of this ethical issue. There are also some practical issues, one of which is that if you make a tissue, if you make a heart for somebody, for example, it has to be a match for that person or else he'll reject it, right? And you've heard, I think, of these advertising campaigns in the city, often for trying to find somebody to donate blood, for example, or bone marrow for somebody who needs a transplant, and they can't find a match, so they go through millions of people. And the reason for that is that there are alleles called HLA, or human leukocyte antigen alleles. These genes are on chromosome 6, but I don't want you to really pay too much attention to this slide, except that there are more than 29 million different combinations of HLA. So in order for somebody to have a perfect match, you'd have to have sort of 29 million options, unless you're dealing with family members, because then you've got a 25% chance of having a perfect match between brothers and sisters. Okay. But you can imagine that you can't possibly get enough human embryos to be able to match everybody. So that's a practical issue, that's a problem. And researchers have recently been able to get around that problem by making what are called induced pluripotent stem cells. So these cells are derived from any tissue in the body. So you can take a piece of skin from somebody who needs a transplant, and just by injecting these 4 genes into the cell, these are called transcription factors, the cells can be reprogrammed so they become very similar to embryonic stem cells, and then you can direct them into any tissue. And the advantage of doing this is that you're taking the starting tissue from the patient, so it's a perfect match for him. So you've eliminated that problem of the 29 million different possibilities for matching. So these cells are called iPS cells. They resemble embryonic stem cells. And as I said, they solve the matching problem. But there's still one more problem with embryonic stem cells that is significant. And that's why you haven't seen any clinical treatment involving embryonic stem cells so far, even though they were discovered about 20 years ago. And the problem relates to one of the advantages, that is that you can grow these cells indefinitely. So you can keep passaging them. They'll keep dividing forever. But what happens after about 20 cell divisions is they start to adapt themselves to this in vitro culture condition, and they gain extra chromosomes. So this is a karyotype. This is showing the chromosomes in an embryonic stem cell line. And there should be 46 chromosomes, right? So this cell line has an extra chromosome 17 and then an extra chromosome 12 that developed after about 20 passages. And this is now a cancer cell. Okay. So that's what happens with embryonic stem cells. That even if you differentiate them into tissue, you can't completely differentiate them, so they still have this propensity to develop into cancer over time. And that's why there are no embryonic stem cell treatments today. That might be overcome in the near future. People are really working on this with the induced pluripotent stem cells, but so far that's still the major problem. So all the treatments that are available right now are using adult stem cells. So an adult stem cell is a cell that is found in any tissue that's already differentiated. So our skin has stem cells that makes new skin all the time. Our colon will have stem cells that will make new cells all the time. The retina of the eye has stem cells. So all the tissues that are differentiated have stem cells that will make that specific tissue. And that was the thought for many years, that these adult stem cells are stuck in that one pathway. But now we know that we can actually make various tissues from these stem cells. And the classical adult stem cell is the blood stem cell that's found in bone marrow. So this is the only stem cell so far that has been capable of replacing an entire organ. And you've all heard about bone marrow transplants. They've been around for 60 years. So if somebody has leukemia or cancer of the blood, the white cells in the blood, those cancer cells are in the bone marrow. You can give that patient radiation or chemotherapy to destroy the cancer cell, but you also destroy the bone marrow. And then you replace the bone marrow by putting in bone marrow from a donor that matches. And that regenerates the bone marrow, makes new blood cells again. So we were interested in these blood stem cells. The classical source, as I told you, is bone marrow, but these are also found in umbilical cord blood. And there are a lot of advantages to umbilical cord blood compared to bone marrow. First of all, it's completely painless and safe because the baby's born, and so the baby's safe. The cord blood is present in the umbilical cord. You can just drain it out with a needle, and we can get about this much blood from the placenta and the cord after the baby's born. And this would typically just be thrown away in the garbage. So we can collect these cells. The other really interesting thing is that the reason we can collect these blood stem cells is that in the baby, inside the womb, all these stem cells are in the circulation. They're not in the bone marrow. So there's literally within an hour of birth, these cells home in on the bone marrow and they form all the blood-forming elements, the platelets and the white cells and the red blood cells. So at the time of birth, we have the ability to collect the blood that's left in the cord, and all those stem cells are still in there because they haven't gone to the bone marrow yet. And when we actually use these for transplant, it's the same thing. We just put them into a vein, and they get into the circulation, and they automatically home in on the bone marrow, so they actually go to the bone marrow. So it's a really simple method of doing bone marrow transplant. The other cells that are present that have been found more recently are actually in the tissue of the cord itself, and they're called mesenchymal stem cells. They're also multipotent. So we're now storing not only cord blood in a bank, but we're also storing pieces of the cord to get mesenchymal stem cells. And they will also, they're also self-renewing, they're also multipotent, and they can be derived into various tissues. So I just wanted to tell you about some of the research that we're doing with these cord blood stem cells, because as I told you, we found that they are also multipotent. We can direct them into various tissues. So this slide is a section through the spinal cord of a man who died of a spinal cord injury. Most of these patients are young men because the accidents are the major cause of spinal cord injury. And in a human, what happens is the cord isn't cut in half when you have an accident. It's usually just crushed. And you can see here, these spaces in the spinal cord are where the primary injury occurred, and those cells actually died, so there's the cyst left in the cord. But you can see there are other areas where there are neurons intact, but they're pale in color compared to this blue staining here. So the blue is staining for myelin. It's a sort of a fatty type substance that covers the nerves and insulates them, so it allows electrical impulses to pass through. So even though this man had a spinal cord injury and was paralyzed, he still has neurons that are intact, but the electrical signals won't pass this site of the injury because these cells don't have this myelin sheath on them. So the cells that make myelin are oligodendrocytes. So this is a cartoon showing oligodendrocytes in blue. So they form the myelin sheath on the axon of the nerves, the nerve cell. And then the red cells are astrocytes, so they just sort of support all the other tissue in the nervous system. So the nice thing about oligodendrocytes is that you only need one of them to myelinate about 10 nerves or so. So you don't need a lot of them to restore that myelin. So we decided to try to make these cells from umbilical cord blood stem cells. Dr. Rogers in my lab at Mount Sinai did the work. And he was able, just by adding vitamin A and some other chemicals to the cord blood cells, make cells that look like nerves. Here's some cells that look like astrocytes, those cells that support the nervous system. And then here are the cells that we were interested in. They're oligodendrocyte cells. So they look like oligodendrocytes. They have all these multiple projections that would go to the various axons to myelinate them. And we could prove that these really were oligodendrocytes because we stained them with an antibody against human myelin. And you can see that the cytoplasm of these cells is packed with myelin. That's the red staining here. So we did an experiment in rats where we actually we mimicked a human spinal cord injury. We made a crush injury in the spinal cord. So the rats had paralyzed hind limbs. And then we injected either these cells or some fibroblasts for a control group just to see if we could repair that spinal cord injury in the rat. And what we found was after 6 weeks, the rats who got the cord blood stem cells had a significant improvement in their hind limb function. They could almost get their hind limbs underneath them to bear weight, whereas the control group wasn't able to do that. So this is just one example of what could be done with a so-called adult stem cell that people believe wasn't able to do anything else except make blood. And the other beauty of this is we've done probably thousands of transplants of these cells, and we've never seen a tumor. So these adult stem cells seem to be very safe from the point of view of forming cancer. I just want to show you a couple other pictures. This is a picture of a human blood vessel that we formed in a rat leg, where we tied off the femoral artery to make the leg ischemic or have low oxygen. And then we put these cord blood stem cells in, and they were able to actually form this human blood vessel. And here's another picture of a very pretty picture of the vessel. So all the staining, the green and the blue is for myosin and actin, and the red is for I've forgotten what the name, lamina, I think, lining the blood vessel. And then finally, I just want to show you this picture. This is what the cord blood stem cells look like initially. They're round, they look like blood cells. And then when you stimulate them for about 3 weeks with various growth factors, they change their shape. They start to look flat and spindle shape. And these cells actually now start to look like pancreatic beta cells or pancreatic islet cells. And again, when we stain these with an antibody against insulin, you can see that the cells are packed with insulin. It's a green staining. So we were actually able to make beta cells that could make insulin for somebody who's diabetic. Again, because there's so many, it's so easy to get cord blood, there are probably over 1 million cord blood samples banked worldwide in public banks and at least 1 million in private banks worldwide. So it probably would be possible to find a match for somebody who has diabetes and to make beta cells for them that could be put into the liver to produce insulin. So just to summarize then what we talked about, so embryonic stem cells are derived from a human embryo. There are problems ethically because you have to destroy an embryo. There are problems because they form tumors. And there are problems because you have to match somebody, but that has been overcome by the development of induced pluripotent stem cells, which are the same as embryonic stem cells, but they match the patient directly. And there's lots of research right now going on to see if they can get around that tumor formation issue, because if that's possible, this would be a really great source of tissue therapeutics in the future. In terms of adult stem cells, they're found in all tissues, but the ones that we've been focused on have been umbilical cord blood, both the blood stem cells as well as the mesenchymal stem cells, which can both be differentiated into all sorts of different tissues. So this work's been really exciting. We've been doing it for about 10 years now at Mount Sinai, and be happy to answer any questions that anybody has. Thanks very much, everybody.
Anybody have any questions?
Yeah.
You were talking about the umbilical cord, the placenta. The stem cells gathered from that in the blood, would you be able to use those cells? Would it have to match the?
Yeah, so that's a good question. So if we stored somebody's cord blood, who could it be useful for? So obviously it would be useful for the person that we stored it, who we collected it from, because it would be a perfect match for them. We talk about family banking right now because that sample is good for probably everybody in the family. What I didn't tell you is that these blood stem cells from the umbilical cord are really immature cells. They haven't developed any kind of immune surveillance yet. So you don't have to have a perfect match to use them. You can be out 2 or 3 of those HLA alleles, and they'll still engraft and make bone marrow. So right now for bone marrow transplant, if you have a cord blood sample from a family, it'll be a perfect match for 25, like a quarter of the siblings. We have lots of siblings. It's about a 50% chance it'll be a perfect match for the parents, and probably a close to 100% chance that somebody in the family will be.
Hi, I'm just curious, when you bank cord blood, what's the lifespan of that blood?
Okay, so the blood's stored in liquid nitrogen. So what we do is we take the blood from the cord, we centrifuge it, so we separate the red blood cells from the white blood cells, and the stem cells are in the white blood cells, so we only have a small volume of white blood cells, and then they're frozen in liquid nitrogen. And the thinking right now is that they probably will last pretty much forever in liquid nitrogen. The rate limiting step is the container that they're in. As long as the container is intact, the stem cells will be okay. And so far the longest cells that have been frozen have been 27 years. Hal Broxmeyer in Minnesota was the guy who came up with this whole idea that you could use these cells for bone marrow transplant. So he's been thawing a sample every year for the last 27 years. They're still good.
Wow.
How soon after patients with leukemia are treated with radiation do they give them stem cells?
Okay, so did you hear the question? It's how soon after somebody with leukemia is treated with radiation or chemotherapy do you give them the stem cells? So the way it works is they first of all try chemotherapy because some people you can knock out the cancer cells and maintain their bone marrow, and so they go into what's called remission. And sometimes you can cure people with chemotherapy, but in cases where the patient keeps relapsing, then they keep bumping up the dose of the chemotherapy. They add radiation and chemotherapy. And essentially you knock out the cancer, but you also knock out the bone marrow. And the problem with that is that those patients then are liable to get infections because they don't have any white cells left to defend them. And they can also bleed because they don't have any platelets. They don't make any red blood cells. So they need multiple blood transfusions. With the bone marrow transplant, the bone marrow, they find a donor before they do all that chemotherapy and everything. And then they put needles into the sternum or the iliac crest right here to suck out the bone marrow. For a bone marrow transplant, you need 1.5 liters of bone marrow. So the patient has to have a general anesthetic. The donor has to have a general anesthetic. And you have to suck all this bone marrow out, which is very painful otherwise. But that bone marrow is then put back into the marrow cavity, and it takes about 2 weeks to engraft, so make new blood cells and platelets and everything else, white cells. With cord blood, it's totally different because you just collect it from the cord, you just intravenously infuse it, as I told you. And you do it right after the chemotherapy has knocked out all the cancer cells. And the difference is there aren't as many cells because we only have about 100 mils rather than 1.5 liters. So it takes about 4 weeks for a bone marrow transplant to engraft from a cord blood sample. So that's one of the things we're working on right now is trying to expand those cells in vitro in a dish to get more cells so that we can shorten that period of time that you have to keep the patient in isolation where they could get an infection or have a hemorrhage.
I don't know if I missed this, but for the umbilical cord, when you take the tissue, can that be influenced to be put in anybody, or is it the same sort of thing that you have to have a perfect match?
No, so the question was when we take the cord blood or the tissue from the cord, can that be used for anybody, or do you still have to have a match? And the answer is yes, you still have to have a match. But it's a lot easier to get thousands of those samples because everybody, there are millions of deliveries around the world every year.
I know that dentists can pay to have their cord blood banked. For the dentists that don't decide to do that, do they need to give consent for research to be done on that cord blood? Do they have to donate it back, or can you just use, if anyone doesn't bank it, can you just use their cord blood?
So right now there are two types of banking. There's public banking, which just started in Canada. The federal government has now put aside $48 million for public banking, which means that anybody can donate their placenta and cord blood to the public bank where it's stored and can be used for anybody in the country. There are only about 4 hospitals right now that actually will accept those samples, so not everybody can actually bank in a public bank. For the private banking system, you actually pay to bank your own son's or daughter's sample. And it costs about $3,000 for 18 years of storage for the collection and storage. And sorry, what was the rest of the question?
Just in terms of whether or not for consent, so if
Oh yeah,
you
sorry,
want
right.
to, do they have to consent that you can use it for research?
Yeah, so any kind of research we do has to go through an institutional review board, and there has to be a consent form. So the patients would have to sign a consent allowing us to do the research on their blood if they didn't want to bank it or didn't want to donate it to a public bank. Yeah.
So I'm just curious, how well do these stem cells survive in a body infected by like an autoimmune disease or something like that? And can they help cure autoimmune disease?
Okay, that's a really great question. And that really pertains specifically to diabetes, right? Because type 1 diabetes is considered to be an autoimmune disease, so the body actually has destroyed the islet cells. So you can make all the islet cells you want, put them back in, they're going to get destroyed again. So you have to address that autoimmune problem. And one way to do that is by doing the mesenchymal stem cells at the same time as the tissue cells that you're putting in. So mesenchymal stem cells have a very unique property in the sense that they can sort of reset the immune system. So they will reduce the autoimmune problem. They'll also reduce a condition called graft versus host disease, where you do transplant of bone marrow or something. And because it's not a perfect match, the bone marrow actually rejects the patient. It's not the patient's rejecting the bone marrow, it's the other way around, because you've now put in a new immune system, and it's rejecting the patient. And that's really, it's a terrible condition, and a lot of patients die from that. So it looks like mesenchymal stem cells can actually suppress or dampen down that autoimmune condition and prevent graft versus host disease, and it might be useful for transplants as well in patients with diabetes. And actually, these cells may also work for conditions that are strictly autoimmune, like multiple sclerosis or lupus, systemic lupus, or juvenile rheumatoid arthritis. So there's lots of conditions that might be treatable just by trying to influence the immune system with these cells. Yep.
Do you have to take the cord blood immediately after the baby's delivered, or will the cells develop?
Yeah, so we collect it immediately, right at the time of the delivery, but we have 72 hours to process it. So it can be, we collect samples from all over Canada, actually. They're sent by courier to a central lab out by the airport, and the cells are processed at that point. So they're kept in a shipper that maintains a constant temperature. But we have about 3 days to process them, so it's not urgent, but you get them immediately.
All right.
Yeah.
Thanks very much.
One more.
No? Sure? Okay.
Okay, so great questions. Thanks very much, everybody.
Well, thanks very much for coming in. I appreciate your time.
Pleasure.
This relates really well to what we've been talking about in our science classes, and we've got a little gift for you for coming in and taking time out of your day. Thanks very much.